Great. Thank you, Tommy. Thank you very much for the introduction. Always very gracious. Thank you for the invitation as well. Always great to be at BIF this year in particular, being able to finally be back in a room with other people and face to face. It's really great. So I'm going to talk a little bit about our research on the carcass and meat quality. And what we did in this project was actually trying to look at pleiotropic regions for carcass and meat quality. And this is on crossbred cattle, as I'm gonna get to it in a second. I am going to point out the fact that most of this work was done by my group, which was in front with Fernanda Rezende. She still is a postdoc in my lab, but also graduate students and other faculty. So when we talk about carcass and meat quality, first of all, I don't think I need to convince anybody in this room that these are very important, and they should be important attributes for the beef industry for a number of reasons. First of all, because they are the ones driving profitability and consumer demand, but also because they can be improved. So we've seen, and we are going to see a little bit more, a few more heritabilities in this study, but these are traits that have a good heritability we do have the ability to improve if we want to. But in the same time, I would also argue that they are probably even more important when it comes to crossbred cattle. And you get into subtropical, tropical regions where you do have both indicus influenced cattle because of the adaptability that they bring to the table. But these type of crossbred cattle, they do tend to underperform when it comes to carcass and meat quality compared to their purebred counterparts. So also on top of that, we talk about carcass and meat quality. As we've seen already a number of examples, they are challenging, especially on the meat quality side, quite expensive to measure. And also we can only do it late in life. Meat quality, obviously, after we harvested the individual. And so because of that, because of the challenges, because of the fact that maybe phenotypic selection is not viable for a number of these traits, we are really looking at genomic selection to help with that. But I think that before we can embark and say, okay, let's start selecting for these traits, we do need to understand the correlations and the relationships that exist between these traits. And so that's why we wanted to look at pleiotropic regions, the pleiotropy, the correlation, genetic correlation that exists between carcass and meat quality traits. And again, this is in crossbred cattle. Again, focusing a little bit more on the carcass and meat quality, how do we describe this type of traits? Well, both of them are complex concepts. And what I mean by that, there is not one single trait that I can say, if you measure this is what describes carcass, or this is one trait that you can use to describe the meat quality. So I do have a bunch of component traits that I can use all together to describe the carcass or the meat quality, but then each individual component trait is also complex in nature. So each one of them is a complex trait. And you know what I mean, controlled by thousands of genes and also under the environmental influence. So for example, if I take carcass quality, I could use these component traits to describe it, right? I can use hot carcass weight, marbling, fat over the ribeye area. If I look at meat quality, first of all, we kind of distinguish between a visual meat quality, and there I can use color, texture, firmness. Those would be the component traits that I can use to describe the visual meat quality. And then I could also describe a sensory meat quality by using traits that we usually measure in a taste panel or consumer panel, and those are juiciness, tenderness, connective tissue, flavor, and off flavor. And again, because most of this, especially when you get into the meat quality, very expensive, very difficult to measure. We don't really have a lot of records, right? That they would be perfect candidates for genomic selection, but we do need to understand those relationships which are behind them before we can start doing that genomic selection. In terms of the research population that we use for this study, so at University of Florida, we have what we call the multi-breed herd. This is a Angus by Brahman herd. It ranges, it ranges anywhere from 100% Angus to 100% Brahman, and it has all the crosses in between. So great population to study either the purebred populations, but also all the crosses in between. We have about 2,000, so almost 2,400 animals phenotyped for carcass and meat quality traits in this population. We do have the pedigree, and we use the pedigree for this analysis, about 4,000 animals in the pedigree, 415 sires, and almost 2,000 dams. In terms of phenotypes, we had almost 2,400 steers with carcass and meat quality data. You see all the traits that we had available to us on that list. Some of the traits are in white, some of the traits are in orange. So what we did, we actually ran a factor analysis first and looked for percentage of variance which is in common. So for example, hot carcass weight and rebuy area, they share a lot of the variance in common. And so we said we are not going to use both of those traits in our analysis, we are going to pick only one. Similarly, marbling and fat over the rebuy share a great percentage of variance in common. We picked only one of them. In this case, we picked marbling. So the traits in orange are the traits that we kept in our analysis, and then the ones in white, we did not. So for carcass, we actually ended up with just marbling and rebuy area. And also the reason we picked the marbling and rebuy area, we thought of them as being economically important traits. If we are going to look at including some of these traits in a selection scheme, those would probably be the ones that we would be more interested in including in that selection. So marbling and rebuy area for carcass. For the visual meat quality, we actually ended up keeping all three: color, texture, and firmness. And then for the sensory meat quality, we kept juiciness, tenderness, and flavor. We had about 1,000 animals genotypes in this data set with the functional chip, that's the 250K. And quality control, you see the numbers there, that dropped to about 221,000 SNPs overall. So I'm gonna spend a little bit of time on heritability's phenotypic and genetic correlation. So you know this type of table. I know there are a lot of numbers. We'll go through some of them in a second, but you have on the diagonal, you have heritabilities. And then on top, you have phenotypic. Above the diagonal, you have phenotypic correlations. Below diagonal, you have genetic correlations. On this slide, I want you to focus only on the diagonal. So if we look at the heritabilities that we've got for these traits, again, remember, this is a crossbred population, but pretty moderate to high heritabilities for the carcass traits. 0.49 for marbling, 0.53 for rebuy area, 0.43 for tenderness. And then low heritabilities for the meat quality, either sensory or visual. Very similar numbers that everybody knows, so very similar to other numbers published in other populations. A number of phenotypic correlations that I'm gonna go through. So this was the first one that jumped at us, and that's the rebuy area. So if you look, this has a consistently low, very low phenotypic correlation with all the other traits, all the other carcass and meat quality traits. So that's the rebuy area. But we do have some medium and positive correlations among, again, phenotypic correlations. For example, marbling versus tenderness and juiciness, tenderness with juiciness and flavor, or juiciness with flavor and color. And then some negative medium correlations, but these are still favorable. So although they are negative in as a number, they are favorable correlations. So the first few traits, a higher number is good. the last three traits, color, texture, and firmness, a lower number, it's favorable. So still favorable correlation, marbling with firmness and juiciness with firmness. If we move on to the genetic correlations, we do have medium to actually pretty high genetic correlations. All of these are favorable. So marbling with a number of other traits, pretty high genetic correlations. Tenderness with juiciness and flavor, again, pretty high, strong genetic correlations there. And the meat quality, the flavor, texture, and firmness, very strong and favorable genetic correlations. So yes, yeah, maybe, if we'll look at some of the genes which we found to be related or underlying these traits, and I think that maybe does make a little bit of sense. If you think of maybe the muscle fiber, and either in terms of texture, how the muscle fiber diameter or would impact the texture, and maybe some of the flavor as well. That's, yeah. So these are the numbers that we get, and we can dive a little bit more into why would we have those strong genetic correlation in a second. And then I left this genetic correlation for the end, actually. A very important correlation between marbling and tenderness, because this, again, as I said, two economically important traits, right? That number of 0.21, it's lower than a lot of other estimates in other populations, but it is a very similar number to what we found in both indicus influenced populations. So, and that's probably suggesting, or maybe it's confirming, that maybe we do have a special relationship between fat and tenderness when it comes to both indicus or Brahman influenced cattle. So looking at those heritabilities, genetic correlations, we felt that there is enough there that we should go ahead and try to look for pleiotropic regions. Why do we have these strong, maybe genetic correlations among these traits, and can we pinpoint some of the regions that are going to controlling these different traits? So in terms of mapping these pleiotropic regions, what we did, we took one trait at a time, and we ran weighted single-step gplot. We used actually not individual SNPs, but 1 megabase SNP windows. And then for each trait, we actually used all the other traits as covariates, and I'll explain why in a second. We identify, so we call the window as associated if it was explaining more than 1% of the additive genetic variance for a particular trait. And then we looked across all the traits, and if we had windows that overlapped on 2 or more traits, those were the windows that we called pleiotropic. So this is a nice example. I picked only 3 traits just to give you an idea of how those pleiotropic regions look like: marbling, ribeye area, and tenderness. And you see everything in green. Hopefully you see it on that slide. Everything is in green that goes above that red line is going to be a pleiotropic region. So explaining more than 1% of the additive genetic variance in 2 or more traits. So let me explain what these pleiotropic regions are. So a pleiotropic region would be a genomic region that would have a direct effect on multiple traits, right? So I have a genomic region which is controlling marbling, it's controlling ribeye area, it's controlling tenderness. Those were the regions that we were trying to map in this study. There are other type of effects, for example, that we would call indirect effects, where we have a genomic region that has an impact on marbling, for example. But then we do know that marbling has an impact, or it influences our perception of tenderness. And so even though you can see the genomic region out there doesn't really have a direct effect on tenderness, when we do the analysis, we would find an effect which would be an indirect effect coming through the marbling. But we were interested in mapping the true pleiotropic region if a genomic region is controlling directly multiple traits. And so that's what we did by fitting all the other traits as covariates in the model when we ran each trait. So trying to account for those indirect effects and try to get and map only the true pleiotropic effects. These are the windows and the chromosomes where we found this pleiotropic region. So we had 5 chromosomes. You see there on top the chromosome, the exact window that was influencing the traits. And then below you have the trait that we're controlling. And in parentheses, you have the percent of direct additive genetic variance controlled by that particular window. So you can see we had 6 regions across the genome, explaining quite a bit of the genetic variance in those traits. We also had 3 others, which we call them suggestive pleiotropic regions. They did not reach the 1% threshold, but they were anywhere between 0.9, explaining 0.9 to 1% of 2 or more traits. I wanted to focus just on 2 of them. So the first one I'm going to talk about is this region on chromosome 5. And this is the region that was actually controlling most of the traits we had in our analysis: marbling, rebuy area, tenderness, juiciness, texture, and firmness. And you can see that it's actually explaining quite a bit of the genetic variance in each one of those traits. So very important region in our mind. We try to look for genes which are in this region. There are a lot of them. I only put 5 so I can fit them on this slide, and maybe you are able to read a little bit about each one. But when we look at all of the genes in this region, a lot of them, if not all, are really related to muscle development, myoblast proliferation, lipid metabolism. So very easy, if you look at those processes, very easy to maybe say, okay, well, then it makes sense that that particular gene, if it has an effect on all these biological processes, could influence marbling and rebuy area in the same time. Maybe not that obvious, but again, as I was saying, maybe you can see how they could also influence tenderness or texture or firmness, because if I do have a gene that is maybe controlling muscle development, right, that impact on muscle fiber diameter and density, we could probably perceive some of that when we look at firmness or the texture of the meat. So I think we can make those connections that way. I also wanted to focus a little bit on the region on chromosome 29. And that's not because this one is related to a lot of traits in our model, but because this particular window on chromosome 29, right, has a particular space in the heart of any meat scientist in this room, right? Do you know why that is? Let's see, how many true meat scientists are there? I'm sure that Dr. Perkins knows. But when we looked at this region, so first of all, it only controls tenderness and flavor. And these are the genes which are related or we found in that region. There are about 19 annotated genes. All of them are structural proteins. And I'm sure that if you had some time to look through them, you know why I said this is an important region for animal scientists. But that's because calpain is on that list. So calpain is really, one of those key candidates that we still have for when it comes to tenderness. Is probably the most important candidate gene that we have. Although we still do not have a functional mutation identifying the gene, we do know that is involved in the tenderization process because of the role it place in the postmortem proteolysis. But a lot of other genes on this list, you know, they are, as I said, they are structural proteins. So they do play a role in this standardization process because they could allow the attachment of those cytoskeletal proteins of the membranes, the extracellular matrix proteins. And maybe that's why, you know, it's really hard to get to the bottom of what exactly in this region is what causes that effect in tenderness. And it may not be only the calpain gene in that region, because not only that you have all these genes that each one of them makes sense to be related to tenderness, they do co-localize, so they are all in the same region, but they also interact with each other. And there are a lot of studies that showed co-expression of their relationship, which makes this region really tough to pull apart and really try to get to the bottom of those signals. We also did a functional analysis, so trying to get a little bit more insight into the role of the genes that we found in these pleiotropic regions. What do they do in that muscle? So we did a gene ontology and pathway enrichment analysis, and I'm going to focus on only two pathways that we found enriched in this analysis. The first one is a calcium-related process. Again, makes sense, right? Not very surprising. We knew that calcium and potassium are going to be involved in the tenderization because of the proteolysis that takes place. So that is a pathway where a lot of the genes in these pathways, they pop up in all those pleiotropic regions. And then the second pathway, which was enriched in the pleiotropic regions, was the cell signaling and the cell-to-cell adhesion. And again, having structural proteins, you know, which are involved in the proteolysis, either during or after the aging process, you can see how they are involved in the meat quality that we measure here. So I think we are, you know, it helps putting that all those pieces into the big puzzle and trying to figure out which are the genes which are important and how do they work together to give us the phenotypes that we are interested in. So what we did, again, identified a number of pleiotropic regions, and I only showed you the really big ones. So we put a threshold at 1% of genetic variation in 2 or more traits. So that's a pretty high threshold, which allowed us to really try to look into those regions and make some sense of it. But there is a lot more underneath, and we can lower the threshold and we get more genes and we get more work to do. But the 5 genomic regions were on those chromosomes, again, explaining more than 1% genetic variance of more of 2 traits. All in all, if we put all the genes in these 5 pleiotropic regions, there are 317 genes. So still a lot of work to go through them and figure out what they do and how do they play together and how do they influence all of those traits. But as I said, most of them are either cytoskeletal proteins, are important factor in this muscle proteolysis, and also just key players in cell growth, muscle development, lipid metabolism, and fat deposition. So I think we gain a little bit more information that we need to try to really get, and we are not there, but it just helps in our quest of trying to understand the complex architecture of this type of traits and the genetic correlations moving forward to try to maybe incorporate that in genomic selection moving forward. So with that, a lot of students to thank and a lot of financial support, quite expensive work there, but I'm gonna leave you with those pictures and take any questions if you have. Yes.
Previous slide explaining more than 1%.
Yes, of 2 or more traits. So if they explain, so pleiotropic region would be a region that explains some genetic variation in marbling and tenderness. So we looked for the same region explaining genetic variation in more than 2 traits. That would be a pleiotropic region. Well, maybe one.
Is 2% really important or 3%?
I think actually 1% is a pretty good threshold.